I'm John Dunne at NOAA's Geophysical Fluid Dynamics Laboratory, and I'm going to be sharing some thoughts on the use of ocean biogeochemical observations in global retrospective analysis and seasonal to decadal prediction. A roadmap on understanding and predicting ecosystem change starts with field observations, but includes a broad suite of factors, including emissions, scenarios, greenhouse forcing responses, physiology and ecology of the marine system, as well as the environmental context in marine provinces, which are informed by laboratory studies, theory and synthesis, high-performance supercomputing, as well as statistical modeling analysis, all to arrive at a characterization of marine ecosystems. In this talk, I'm going to try and explore the breadth of the contributions of these factors and how ocean biogeochemical ARGO can serve our increased understanding. I'll start with some current uses of ocean biogeochemical observations and global models, explore the partitioning sources of uncertainty, give some background on GFDL's global climate and Earth system models, explore some aspects of detection and attribution of ocean biogeochemical variability and change, as well as Earth system predictability and prediction on seasonal to decadal timescales, and end with some case studies of the value of ocean biogeochemical ARGO for modeling. One current use is this new ECCO analysis by the MIT group, showing the ability to ingest observations in a fully comprehensive and internally consistent representation of ocean biogeochemistry, which compares well with other reanalysis products. Another example for model initialization is the application of advanced statistical methods such as the Bayesian neural networks used by Bittig et al. As an alternative to static climatologies to characterize biogeochemical variability in optimal relationships between the biogeochemical parameters and the physical context. Current global Earth system models use observations in a variety of ways. For initialization, they use climatologies from the World Ocean Atlas and Global Ocean Data Analysis Project, run out with pre-industrial forcing before historical and projection simulations. Process representation can be combined with theory during algorithm development if a process is captured and the mechanism is understood. They use observations for calibration during model development and tuning to reproduce, for example, a robust mean or pattern that emerges from the observations. And finally, during falsification phases of model evaluation, observations are used to refine structural choices for future development. Now moving on to partitioning sources of uncertainty. Hawkins and Sutton, 2009, derived a framework for narrowing uncertainty through the exploration of model or structural uncertainty in contrast to the uncertainty associated with scenarios applied to models and the internal variability that is represented within the models, such that the internal variability drives the highest uncertainty initially in these models, whereas model structure contributes on medium time scales, ultimately to be dominated by scenario uncertainty on longer time scales. Now, as Lewandowski et al. Showed in the context of the CESM model, at the global stage, these the internal variability uncertainty shown on the left and the structural uncertainty on the right were each found to be smaller than the signal in terms of the global scale response. So as you can see, that on the left, the uncertainty starts off quite large, as the red line in panel E, but quickly diminishes over the decades as the scenario uncertainty becomes dominant. In panel F, you see that the situation isn't quite as clear when you come to incorporate the structural uncertainty, as the model uncertainty in panel F dominates initially even over the internal variability uncertainty, and then only towards the end of the century falls into to become a minor contribution relative to the scenario uncertainty. However, when you move from the global scale to the regional scale, in this case, the California Current Large Marine Ecosystem, you see that uncertainty remains throughout the century in the response of this system under climate change forcing, as internal variability in the system and model structural uncertainty prevent easy identification and attribution of carbon fluxes within these models. So there are also a whole series of implicit sources of uncertainty, where the bias in the models is not, if all the models have the same bias, you can't gauge easily the role of that uncertainty. If the resolution of the models prevents the representation of the underlying processes or their relative contributions, there's the question of unresolved initial conditions, limited observations, cases where the model rejects observations because it isn't able to assimilate them into the structures that it's capable of representing, as well as the forcing uncertainty, such as a volcano erupts and predictability is diminished because of it. Now, another one is also in the biogeochemical context, the fact that biodiversity and ecosystem structure and function is not necessarily fixed in time and can undergo structural change. Moving to the background on GFDL global climate and Earth system. For the recent coupled model intercomparison project phase 6, GFDL contributed 2 models called CM4 and ESM4. Both models include an atmosphere that's on the order of 100 kilometers, the CM4 having a lower top than the ESM. The atmospheric chemistry, more comprehensive in the ESM, including ozone. Both included MOM6 ocean with 75 levels, but the CM4 having a 1/4 degree versus 1/2 degree in the Earth system model. Ocean biogeochemistry, much simpler in the CM4 with only 6 tracers relative to 33 tracers, and a much more comprehensive ecosystem in cobalt formulation with more advanced land and the ESM and the same sea ice model. So these models contributed to CMIP6 in a whole variety of different contexts. CMIP6 was a very comprehensive and diverse suite of different model intercomparisons, including a standard description of idealized and historical forcing, the future scenarios, a carbon dioxide removal intercomparison, land use, coupled climate carbon, aerosols and chemistry, detection attribution ensembles, radiative forcing, cloud feedbacks, monsoons, and flux anomaly forcings, all of which are publicly available on the ESGF website. So GFD also conducts a whole hierarchy of biogeochemical modeling for a diverse set of applications. Now, these include the CMIP5 previous generation, 1 degree ocean Earth system models, a exploratory ultra high resolution 10th degree set of CM 2.6 and ESM 2.6 models, a ensemble Kalman filter data assimilation with the 1 degree ocean, including our cobalt model, and as well as the CMIP6 models I described earlier, and then individual exploration of the role of resolution from 1 to 1/8 degree. So these models represent the coupled elemental cycles of carbon, oxygen, phosphorus, nitrogen, iron, alkalinity, silicon, and lithogenic, and how they are coupled and decoupled. And in the case of cobalt, they represent 3 phytoplankton productivity groups, small and large and diazotrophic phytoplankton, as well as small, medium, and large grazers with explicit bacteria and DON, dissolved organic nitrogen pools. In the case of the simplified BLING, biogeochemistry with light, iron, nutrients, and gas, what we do is we set the phytoplankton and zooplankton communities to steady state and solve the equations for productivity as a function of light and nutrient limitation. And these models, we will find the biogeochemistry representation is largely limited by our ability to represent the physical climate. Here shown the evolution of these models over time from ESM to M, which was used in CMIP5, and CM3 also used in CMIP5, having an RMS of sea surface temperature of 1.2 and 1.01 respectively, compared to our latest generation models, where the RMS in CM4 is down to 0.84 and in ESM4 down to 0.68, almost approximately half the bias that we had in our previous generation Earth system model. So across this class of model, we show a comparison of observations on the left for sea surface temperature, pH, oxygen, and productivity, and then the model ensemble mean on the right set of images, showing that these models get the general patterns of and structures of these properties, but to varying degrees of fidelity. So on the right, we have this Taylor diagram approach where the target is this 1 on the x-axis, where going towards 0 means you underestimate the variability. Going beyond it means that you're overestimating the variability. And then the the curve shows the correlation from 1 to 0. So in the case of sea surface temperatures, the models do all fairly well and cluster together. For oxygen, they do less well and do not cluster as well together. And then down to primer production, where they neither cluster together nor do they have a very good correlation with the observationally based estimate. Now, moving on to the application of these models for detection and attribution of ocean biogeochemical variability. Here's an assessment of this ensemble and by Kwiatkowski et al. From last year, showing projections, historical and projected sea surface temperature, pH, oxygen loss, and nitrate and productivity across the range of emission scenarios, and as well as a comparison of the results from the CMIP5 generation models, these RCPs, showing the increase in temperature, decrease in pH with acidification, and declines in productivity and oxygen and surface nitrate. Now, what you'll notice is that while the pH has no shaded area, meaning no observable structural or internal variability uncertainty associated with it, these other parameters become much more uncertain as a function of time. Most notably, the prime productivity, where much of these projections overlap across scenarios and across models. That result is shown as a function of space here in this set of examples where the historical and SSP 2.6 and low emission scenario and SSP 585 high emission scenario are compared as anomalies. So the low emission scenario warms less, and you can see from all the little dots that most of the planet has agreement among the models. The same is true for ocean pH, where there is considerable agreement. But then as you get to questions of oxygen, nitrate, and productivity, there become many areas of the ocean for which there is not agreement in the sign of the change among the different models. And I highlight this because this is an important example of where the ocean biogeochemical algorithm presents an opportunity to look at long-term change and confront models with high quality and robust characterizations of Earth system change. Now, there's to make this point a little bit finer, that the robustness of change in ocean biogeochemistry really is extremely property dependent. I'll point to this recent paper by Sarah Schliniger et al., who explored the time of emergence of anthropogenic signals in the ocean carbon cycle. Now, the upper panel shows the percentage of the ocean area that has emerged, has changed as a function of time from 1990 through the 21st century with a CMIP5 forced GFDL ESM2M. And you see that pH and PCO2 emerge very quickly in the purple, but as we migrate through saturation state, alkalinity, the carbon pump, sea surface temperature, air CO2 flux, phytoplankton biomass, nitrate, apparent oxygen utilization down through productivity and mixed layer depth, the percentage of area that emerges becomes later and later over all of these in this sequential order. This is shown as a map on the lower left-hand panel with the time of emergence of the integrated chlorophyll having changed detectably in some areas very soon, and in other areas not even by the end of the century. On the right-hand panel, you see that the signal in some of these regions is also highly correlated to the time of emergence. That earlier signals become very strong by the end of the century. Now another role of observation model comparison comes in the role of looking at interannual variability. Here's an example where we explored the biogeochemical variability in the California current system. This is just a comparison of the observed climate with a warm ENSO in the center and cold ENSO composite for sea surface temperature and sea level pressure. And in the lower set of panels, the same variables within GFDL's high-resolution ESM 2.6 in climate, warm, and cold composite anomalies. All of that mostly this is due to the rule of resolution, where you can see that the ROM simulation, that's a regional simulation forced by changing boundary conditions, is also capable of showing these regional patterns. And moving on to the biogeochemical significance of this, you see that the Earth system model shows a strong oxygen response and pH response of diminished oxygen in the upper water column and enhanced oxygen in the subsurface during warm phases of ENSO, as well as higher pHs in the subsurface under ENSO. Now, these are examples where the ability to ingest information to the model is extremely limited by the ability of the model to represent the relevant processes. As another example, we have a comparison of the baseline cobalt in this case a regional ROMS configuration, which had a low bias that was not representing the extreme. These are violin diagrams, which are histograms that are mirrored on top and bottom. So you can see the distribution of chlorophyll and the fact that the baseline simulation did not represent chlorophyll values above about 3 microgram per liter. When we added a third size class to cobalt, we were able to then generate these high chlorophyll values observed in the coastal region, but also were led to highly diminished chlorophyll values offshore as nutrients were consumed more closely to the coast. And here's a comparison of these the baseline cobalt and this amended cobalt with the Calcoffee time series, showing that we were able to get this nice chlorophyll bloom along the coast, and a subsurface chlorophyll maximum that agreed better with the observations. So this had consequences for the representation of hypoxia in the region, where because we were able to get productivity focused more on the coast, we were able to represent lower upper water column oxygen values more consistent with the observations. So a success in structural. Now, moving on to Earth system predictability and prediction on seasonal to decadal scales. Now, the predictability question, gets back to this issue of how much memory is in the system, depending on our ability to characterize the initial conditions, and the understanding that small deviations in initial conditions can lead to chaotic behavior in some systems. This in the ocean biogeochemical context, one of the seminal works by Séférian et al. Demonstrated the ability of ocean biogeochemistry models to reproduce the chlorophyll response to ENSO variability when the model was nudged at the surface. It was an Earth system model nudged at the surface with sea surface temperature variables. Here you see the dotted line is the free model, the black line is the observations, and I'm sorry, the black line is the nudged model, and the orange line is the observations. Similarly, in the lower panel, the green line is the observations, dark solid black is the nudged model. They were Séférian et al. Were also showed that there's a great deal of persistence in the ocean biogeochemistry that exceeded the temperature. So looking at this right panel with the lag in years versus the anomaly correlation, you see that whereas sea surface temperature in orange declines in skill early, the nitrate and productivity and iron anomalies persist much longer, and where these blue and red dashed lines are significance levels. So building on this success, that ocean biogeochemical signals might be predictable on seasonal to interannual timescales, Frolicher recently did a study of looking at how much predictability there is within a perfect model framework. How to what extent is the model predictable of itself by taking an ensemble of slightly perturbed initial conditions at 6 time periods in a control run, and looking at how long it took to get these models to diverge into chaotic behavior, such that the initial condition no longer mattered. So they were able to find that the sea surface temperature had areas of predictive skill that had decadal timescales in some regions, like the North Atlantic and parts of the Southern Ocean, and that skill extended also to surface pH, surface oxygen, and net prime production. But that there were other regions that had much lower predictive skill, on the order of 1, 2, 3 years. So they then distributed the planet into a suite of different biomes, and I show this plot to point out that these areas of the ocean behave very differently in their potential predictability. Many of them have seasonal cycles, such as this biome 10, where there is considerable productivity only in the winter season, and that it reemerges each year, and others where predictability declines fairly uniformly without this seasonal oscillation. They then explored the mechanisms of this predictability, where they were able to attribute the skill to the solubility and the apparent oxygen utilization changes in the case of oxygen, and then in the case of the carbon system, a broad suite of factors, including running through sea surface temperature, alkalinity, DIC, and their covariances. Okay. Another recent study compared the relative role of biogeochemical initialization to physical initialization. Now, what they found was that within the first year, these red dots, the biogeochemical initialization uncertainty decreased the skill from 1, and the having only the physics initialization decreased the skill to slightly less, but that over time to the next year, they found that knowing the physical state was much more important to the evolution of the biogeochemical state for both export production and air-sea-CO2 exchange than was the biogeochemical initialization. As knowing the physics but randomizing the biogeochemistry did not negatively influence the ability to reconstruct the evolution of the biogeochemical state. Now, to make a long story short, I quote them: "Our results suggest that initialization of the biogeochemical state does not significantly improve interannual to decadal predictions, which relate to the strong control ocean physics exerts on the biogeochemical variability on these timescales." Here's just another example of this work partitioned into regions, illustrating that the amount of predictive skill varies with region and with season. At GFDL, we found a similar results. In our case, building on GFDL's physical prediction system, and in the absence of ocean biogeochemical initialization, going through the sequence of producing a biogeochemical retrospective, performing retrospective forecasts, and assessing biogeochemical. This comparison between the control initial data assimilation and optimal data assimilation is shown in these Taylor diagrams. Again, so in the case of nitrate, you see that going from the control to the initial data assimilation strongly decreased the fidelity, and only in the optimal case increased the fidelity in both the global and equatorial sections. A subsequent study implementing this model in a retrospective forecast mode was able to demonstrate large areas of the ocean where we found seasonal to interannual forecast skill with respect to satellite comparison with satellite chlorophyll. Here, some of these regions highlighted where the signal reemerges in subsequent years, and is strong relative to persistence in the dots. So here's a closer look at what that color looks like, where we compare the model and the OBS for the equatorial region with the correlation of 0.78. We were also able to connect the correlation skill to estimate of fish productivity in some regions with a 0 to 1 and 1 to 2 year lag. Similarly, we were able to explain the multi-decadal variation of oxygen at the Calcaffe area, where the model's internal evolution of oxygen was captured through this long-term variation in the physics. Now, one of the emerging paradoxes in climate science is the signal-to-noise ratio paradox, where models appear to be able to predict the real world better than they are able to predict themselves, such that while you might expect the perfect model experiment to provide the upper limit to the predictability, since the model represents some aspects of the real world, in many cases, such as the North Atlantic Oscillation, there is a sense that the sources of variability in the models are more chaotic than those in nature. This has great potential that the tools that we're developing, as they become more mature, may be even more relevant to the prediction question than we currently appreciate. And finally, a few case studies of biogeochemical Argo value for global modeling. One of them is using data to identify global modes of variability. Here's an example of the hot time series, where we see a Eulerian section in time. Another is the typical process study and transect cruise, where you get a picture in space and some time, whereas the past biogeochemical observations have been limited to a few global sites and surveys where sampling is biased against bad weather and against winter conditions. As in core Argo, biogeochemical Argo will move model observation comparison from the decadal time scale of global synthesis to monthly or better snapshots of variability, allowing key new observational constraints and a four-dimensional description that will greatly facilitate detection and attribution by decreasing the degrees of freedom that you have when you only have a time or a space characterization. And a second way is that these forecast models will benefit from reanalysis efforts such as ECCO that can be used for verification and falsification. Another use of data for modeling is in the development of regressions for water mass analysis to constrain initial and boundary conditions for models. Another application of data for models is to compare the resolved variability in observations from efforts like Canyon B that use more sophisticated water mass characterizations and identify those cases of observations that deviate from these null hypotheses, where you find something new that might be relevant for future modeling constraints. Yet another application of the ocean biogeochemical Argo observation is in contextualizing surface PCO2 and chlorophyll constraints. While these observations are relatively vast and powerful for estimating instantaneous air CO2 flux and ecosystem state, respectively, they're challenging to serve as model constraints. It's difficult to know what depth range the measurement represents. While potentially relevant for modeled dissolved organic carbon, PCO2 is only about 0.3%. Similarly, chlorophyll is only part of the ecosystem story. And finally, the mechanisms that drive model observation mismatch are manifold, with winds, temperature, nutrients, lateral and vertical processes all important drivers. Biogeochemical Argo holds the promise of providing the vertical context to these surface observations such that they can be better compared with models. And finally, using data to compare time of emergence anthropogenic signals in the ocean carbon cycles, we need the observations to understand the robustness of the modeled biogeochemical changes. In conclusion, current global ocean biogeochemical models use observations primarily as initial conditions with long spin-ups and state estimation. There are many sources of uncertainty, including structural scenario and internal variability. Mechanisms can be complex to understand and often interact for different results and different models. Models capture many key biogeochemical regional features, however, and many regions exhibit strong seasonal and decadal predictability and prediction skill, even without biogeochemical initialization. As modeled interannual predictability is derived from the evolving dynamics rather than the ecosystem or biogeochemical state. The predictability paradox suggests that many regions are much more predictable than the models currently suggest, and the advent of biogeochemical Argo will provide a multitude of opportunities to fill in the blanks and provide a much more robust context for model observation, comparison, and validation. Thank you.